share. Share. So can you see my screen now?
Yep.
Yes.
Okay. Okay, so these 4 questions I put is just to give you an idea on the last lecture that we discussed the restriction enzyme, the generation of libraries, and in each of the lectures, I'm trying to give you some questions so you can get a flavor of how the midterm would look like. So probably the questions might be a little bit more trickier in the midterm, but more or less, this is how it should look like. And last year in the midterm, I had only questions that has 1 answer. So I will also give you very good instructions a week before the exam and on the exam sheet itself. So could you please go to the questions and okay. My computer is like clicking so quickly. Okay, so what is a genomic library? A genomic library is a collection of genes coding for proteins. It's a collection of the DNA fragments that represent the entire genome of an organism. It is a collection of the messenger RNA molecules, or it's a library of plasmids carrying antibiotic-resistant genes. I really advise you to answer without looking to any other helping machines that you have around. Like, don't look on Google or ChatGPT or something like this, because I just want you to evaluate how much you got from the lecture. There's no grades on this, and you are all anonymous. So Greta, these question answers that I'm making are in principle the practice for the midterm. Which lecture site are we? It should be the same. If I uploaded 2, it should be the same. There should be no difference, Ashley. Okay, so the answer is 2. Very good. It's in principle a collection of the DNA fragments that we got from the genome of an organism. So it's the whole DNA fragment for the whole genome. It's true for the person who answered number 4. It's true that those are inserted into plasmids, but it's not the plasmids only. It's the plasmids with the DNA fragment. So this number 4 is like confusing because it didn't tell you that the fragments of the DNA are in principle cloned into those plasmids. So be careful when you read the answer. What type of DNA ends do restriction enzymes typically create after cutting? Sticky and blunt, blunt and sticky, only blunt, overhangs with random lengths. We are at 36. There's still like a room for 10 people to answer. 42. 7 more people, please. 43. So, in principle, there's no rule that a restriction enzyme would cut more blunt or cohesive. We discussed that it's based on the sequence. It could be blunt, it could be cohesive. So it's based on the enzyme itself. So when I say restriction enzymes, this means the majority of the restriction enzymes that we know, and this means both blunt and sticky ends. Yes, the cohesive ends are more known, and there is much more cohesive end cutting restriction enzymes compared to the blunt end. But the question is the restriction enzymes. Be careful when you're reading the question again. I will try to make the question more clear because I can see that some of you are picking the wrong answer because the question is not formulated correctly. Then the question is, what enzyme is used to join DNA fragments after they are being cut? Ligase, polymerase, endonuclease, helicase. Beta, what was the right answer for the question? I said which one was the right. For the one before, it was both sticky and cohesive ends. So 38 out of 52, they are saying ligase, and as the name indicate, it ligate something, so it stitches two pieces together. Yes, if you want to join two DNA fragments together, you need the ligase to formulate that enzymatic reaction. It's not the polymerase. It's not the endonuclease, because endonuclease is a restriction enzyme, and it's not a helicase because the helicase is the one that unwinds the DNA. So that's good. Good thing that you didn't hear anything about helicase, so no one answered. I would estimated that polymerase and endonuclease could be a possibility that you made a mistake, but that's good. All of you got the right answer. And do I have one more question? Oops, wait. There is one more question. I don't know why it brings me outside. And which pattern of DNA is easier for cloning during DNA, cloning the resulting DNA fragments? So if you cut the DNA and you want to clone it, which enzymes would you use? Would you use an enzyme that make the DNA blunt, or you would like to use an enzyme that make the DNA sticky? So think about the next process. It will tell you which one is going to be better. Ten more seconds. Still have some room for those who are hesitating. Again, you need to give it a shot to see how much you are getting out of the lectures. I'm not sure if you guys are studying the lectures on time. Okay. So the answer is 2. Yes, cohesive ends or sticky ends are much easier for downstream activities, whether you are doing the cloning, the ligation, because the sticky ends can cause the DNA to re-anneal on itself, and then all you need to do is just the ligation. The percentage of ligating a blunt versus cohesive end is like 3-fold less. In other words, if you do an experiment, you have a chance 3 times more to get a ligation and cloning with a sticky end compared to a blunt end. Okay, very good. So let's go to the lecture. And so this guy, so I deleted that one because it's just a mistake in the name. It's only one module. It's exactly the same as the one that I have here. Okay. So if I go back to my PowerPoint and I share the PowerPoint with you. Okay, please let me know if you see my screen.
Yes, we see it.
Gary, I will answer your question at the end of the lecture. Please remind me. Okay, so this lecture up till now, so you can see we are like building blocks bit by bit. So we are able to culture cells, we are able to count cells, we are able to extract DNA from cells, and we are able to extract the whole DNA from those cells. So that we get the whole genome from those cells and look at it on electrophoresis. Probably we can digest this DNA with restriction enzymes, and we can run it on a gel, and we can see how the pattern look like. In principle, you know how to make a gel, and you know how to load your DNA on a gel with electrophoresis and how the DNA migrate from the negative to the positive. So all these steps are already, I assume you guys know how to do it. Now, we discussed in the previous lecture how we analyze DNA by restriction enzyme, and we discussed how the restriction enzymes are used, how we make the reaction, and what enzymes we need to use in terms of which experiment. And as I can see from the questions in the WOOC lab, you guys already got a very good understanding of that. Now, today, we would like to discuss how to separate DNA fragments. So we will start discussing how to separate specific sequences of a DNA out of the whole genome. And we already touched a little bit on this in the previous lecture when we discussed about Southern blotting and RFLP, the two techniques that are mostly used. And in principle, today, I will elaborate more on those two techniques. I want to make 100% sure you know what is a probe, how to design a probe, where to design a probe, and then what are the applications for these techniques. Because in principle, we are just not learning techniques for nothing. We learn the techniques in order to achieve answering some questions. And in this lecture, we will know how to separate DNA based on the sequence, on specific fragments, analyze those specific sequences, and then we will discuss a bit about DNA fingerprinting, because this is one of the procedures that depends on isolating DNA, digesting DNA, running DNA on electrophoresis, and then how you can differentiate between two people or one patient and one person who doesn't carry a mutation. And how can you say that this person is more predisposed to a disease versus another one? So all these applications are in principle what you have learned in the last 5-6 lectures. And finally, I will introduce you to the polymerase chain reaction. I know most probably you have already studied polymerase chain reaction, or PCR. Some of you might not, because I know some of the students in the class have never done molecular biology in depth or cell biology. So we have to go slowly on this. Plus, I will show you how to set up the reaction for a PCR. In other words, what the material you need and how you design the primers and all the steps. Okay. So it's good to refresh your memory about the DNA replication, transcription, and translation. So you all know that when our cells divide, we have a machinery in our body that performs DNA replication so that we have a copy of our genetic material that becomes duplicated. And each of these copies, which should be identical, can be divided and go to a cell out of the two that has been divided, right? Now, the DNA replication takes place in our body at 37 degrees. So we have to find, I mean, it's really a fascinating process that takes place because the DNA is like double-stranded and it's a helix. And it's also supercoiled because it's in a very small site in the cell, which is the nucleus. So in order to replicate that DNA, we have to unwind it. And in the WOOC lab, I mentioned the enzyme helicase, and the enzyme helicase is needed beside other enzymes like topoisomerase in order to unwind the DNA helix. And then if you look at these two helices, one of those will be a template where a DNA polymerase, which comes and start reading this helix and simply add the complementary nitrogenous base. So if you have a T, as you can see here, then it will add an A. And if it's a C, it's going to add a G, a C, and it will continue all around. This happens also on the second helix. And as the process continue, we will have an identical copy, and there is a lot of proofreading processes that happen here so that the DNA is duplicated in a very precise manner. Then we have the process of the transcription, where a copy of a gene or a copy of DNA has been transcribed. Why I'm saying copy of RNA? Because sometimes DNA is transcribed, but it doesn't produce a protein. So it's not called a gene per se. It can be called a non-coding RNA, for example. The same thing happens with the RNA. The DNA has to be unwound, and then the RNA polymerase, in this case, instead of the DNA polymerase, will come and copy the gene, but now for the T is given an A, and for an A, instead of having a T, you will have the uracil, and you all know that uracil is the universal change from the DNA, which is the thymine. The same thing, we will have to do the unwinding. Only one of the two helices is copied so that we get a single-stranded RNA that is transcribed. And we get this messenger RNA, which will be, in principle, translated into protein. And the protein is going to be the functional macromolecule in our cells. So the proteins are enzymes, they are signaling molecules, they are defensive molecules like immunoglobulin antibodies. So proteins are really performing most of the functions in our cells. So why I'm telling you this, because I want to move to a little bit some of the definitions that we will need to understand the polymorphism, which is the major focus of our today's lecture. The polymorphism, in other words, the differences between the DNA sequences of two individuals. So if you get DNA from different human, there is 99 point something, 99.9, I would say, similarity in the sequences. But yet there are still a very small room where these individuals are not the same. So if we look at the DNA that we see here supercoiled in the cells, it is divided into 46 chromosomes, 22 pairs that are autosomes, and the sex chromosomes, the X and Y, XX in male and females, and XY in males. Now, within those homologous chromosomes, the genes reside on similar locus. So whenever you say the word locus, it means a sequence or a gene that occupies a specific position along a chromosome. And as I said, we have two copies of the same chromosome. Therefore, we have two loci for each gene. Now, a simple example is lost like here for you, where here's the loci for the eye color, and therefore this person has a brown eye. Another loci for the hair color, so it's a different side of a chromosome, and in this case, the hair color is hazel. And this can tell you that the location of the gene might be conserved on a chromosome. However, those genes on a particular locus can have different forms. And you all know that we have different eye color and different hair color. And therefore, the difference in the sequences of these genes, we call it a difference in terms of the nucleotides, and therefore we call them alleles. So the allele for the hazel is not the same as the allele for black or blonde. And the same here for the black or the brown. So the sequence of the gene is different. The loci is conserved, but the sequence might be different, and therefore the proteins that are generated can be different, and therefore the trait is different. Now, a locus that has more than two alleles is considered polymorphic. So any of these genes that we are talking about is a polymorphism. So difference in eyes color is a polymorphism, and difference in the hair color is a polymorphism. But for aspects that we will need in molecular biology, these traits are not very useful because they are common. Because you can say 50% of the people are black-eyed, 20% are brown. So there is not really a very good categorization in those traits. And that's why we will need to look deeper in the DNA sequence in order to find better ways to find, to discriminate people based on their DNA sequence. Okay? So I'm not sure if you heard before about single nucleotide polymorphism. And a single nucleotide polymorphism, we call it SNPs. These are very common, and they cause changes in the gene expression. And therefore, the translation and the transcription and the translation will be different for that gene. So you can imagine if you are changing the C into a G, a C into a T, sorry, then automatically the complementary for the C is going to be G, and the complementary for the T is going to be A. So you already created a difference in the RNA that will be transcribed from this DNA sequence. So Mr. A is going to be transcribing a sequence that is different from Mr. B. And therefore, the SNPs can dictate different genes into different alleles. Now, I give you an example here, and this is from a last year lecture in the pathophysiology discussing the mutations in the genetic diseases. So a single mutation that causes the change from a G into A can cause a missense mutation, and therefore you can see that the amino acid that is being transcribed is not serine anymore, it's aspartic acid. You can have many types of these SNPs, like this mutation here if you take C and you replace it by A, you can make a stop codon, and therefore you can block the translation and you will have a shorter version of a protein. Or you can make a change in the whole shift of the protein. So you can see here, it's alanine, isoleucine, serine, tyrosine, phenylalanine. And this one here, after the mutation, you have totally different amino acid sequence. So there's a frame shift. So SNPs are very important to understand. Now, in the previous lecture, we discussed the Southern blotting, and we discussed that if we want to understand overall the whole genome in terms of the restriction digestion of profiling, we have to first extract the DNA, and then we digest with a specific enzyme, and then we will run those fragments on a gel, and then we will transfer them from a gel into a membrane so that we can manipulate them better. And then we can detect those by creating a probe, which is a single-stranded DNA or RNA based on the condition we are using. This probe has to be complementary to a specific site. So each Southern blot you will do, for example, you can't simply run it like this. You will say, we know that this gene can cause this disease. We will have a lot of information about it. We will have a lot of information about the sequence. And then we will guess, for example, that this sequence is the cause where the change in the gene can cause a disease. So we will label this by radioactive material or fluorescent material. And when I say radioactive, it's simply P32. So the phosphorus is going to be P32. So it's emitting light that can burn a film, so that we can detect this probe. Then at the last step, we can apply the probe on the nitrocellulose membrane. And if the DNA that has migrated shows bands, we will know that we are detecting this particular DNA signal. Now, I just want to make sure that you guys are aware that the steps as follow: extraction of RNA, restriction, the digestion of the DNA, running on the agarose gel, and then most probably we will need to do an amplification step in certain cases. I will discuss this sometime. That's why I put it here in bold for you, because it's going to come at the end of the lecture, particularly if the DNA material is not enough. Then the DNA is transferred from the gel to the membrane. And after that, we have the probe that is radioactive, and then we can visualize it. So typically, when you perform the steps of isolation, DNA digestion, and running on the agarose gel, that's typically what you will see, because you have a smear. This smear means you have a lot of DNA of multiple molecular weights. You cannot know what is what here, because your restriction enzyme simply cut it. And you have 40 kilobases, 20 kilobases, 1,000 kilobases, all that. So that's typically what you see on a Southern blot. Now, when you transfer it from the gel into the nitrocellulose membrane by the transfer process, you will end up adding the probe, and you detect only the bands that the probe will bind to. So that's the power of this technique. Now, this is the base. Now we will go to see some of the applications. But before that, let me see some of your questions. Okay. Would it be fair to say that frame shift is not a SNP, given that it also involves adding bases rather than just a change? Well, a frame shift, it can be a SNP, because frame shift can make... Oh, wait. Are you talking about the insertion? Yes, you're right. It's fair to say that frame shift is not a SNP. It's fair to say that. But in other words, where did this come from? It can be an insertion that could be mistake by the polymerase. Instead of adding one of, let's say here, A is AAG, instead of adding AAG, it just added only one A, and then the frame can happen. So it can still be considered a SNP. Okay, then the first application we discussed after a Southern blotting was the restriction fragment length polymorphism. And in this technique, we rely basically only on the restriction sites that exist in the genome of the individuals we are testing. So we took the DNA from a normal person and a person who are acquiring a disease, and then we digested it with EcoR1 enzyme, and then we run the digested material on agarose gel, and then we did the transfer, and we looked at the bands we are getting using these types of probes. So in condition, the probe is covering this area here. So in normal people, we will have a cutting site for EcoR1, while in diseased personnel, we will not have this because it's mutated. And therefore, a diseased person would not have a digestion of the DNA, while normal person will have two fragments of the DNA. So this is the easiest way to detect if a disease is dominantly happening in a population compared to another, by looking at the restriction enzyme, restriction fragment length polymorphism. Now, more applications of this can be done on the population genetics. So there's one example I listed for you here. So it's where you design the probe and how you want to interpret the result. In principle, the procedure and the technique is the same. So on this side here, you can see that we have two alleles, allele number 1 and allele number 2. In allele number 1, we have 3 restriction sites. In allele number 2, we have 2 restriction sites. So the probe we added was at the site where we can have either one fragment. So if we have allele number 1 and you cut with the restriction enzyme, you will have only this guy, which is a smaller size. And obviously, what's happening is when you cut allele number 1, it will be cut into 4 fragments. This part, the part between 1 and 2, and the part between 2 and 3. And then the part from 3 and below downstream. So you have 4 different fragments, but you are not going to be able to see the 4 fragments, right? Because your probe detects only the piece between 2 and 3. And that's why when you cut allele number 1, you see this band here. And it's smaller in size than allele number 2 because allele number 2 doesn't have number 2 here, the restriction site 2. Therefore, the fragment is bigger in size. So when you detect it, it's a little bit higher on the gel. So if you have a homozygote allele number 1, you see only one band. If you have a homozygote allele number 2, you see another band, but it's bigger in size. And if you have both alleles in the genome, you can see one band that corresponds to A and one band corresponds to A2. And therefore, in this type of analysis, you can easily tell by taking the DNA of the parents, like the mom and the dad, you can take their DNA and check which alleles they have. If they have exactly like this, 50%, 50%, meaning have allele A1 and A2, then there's a chance that one of their siblings will have either double A or either double A1, A1, or double A2, A2. And that's what exactly you see. You see that this girl here, she will have A2, A2, because 2 of the alleles of A2 and this boy here will have 2 alleles of A1. So this is how we tackle most of the genetic diseases that we know. And when a woman is pregnant and she does those genetic tests for the known transmissible genetic diseases, that's exactly what happens. We know what are those genes, and we know the probes to identify those genes. And all we need to do is to take the DNA from the mom and the dad, and we perform this type of analysis. Now, you can design the probe instead of sitting on the allele without being cut by the restriction enzyme. You can choose to have the probe within the restriction enzyme. It's like the example we tested here, but it's more elaborated there. So here the probe is going to bind in the sample A on this 5 kilobase fragment, while in sample B, the probe can bind to a restriction site where you have a 2 and 3 kilobase fragments. So automatically, after taking the running, the gel, and the detection at the level of the detection, you can tell that sample A will reveal only one band because there is no restriction digestion that's taken place, while in sample B, because we have a restriction site in one of the two, you will see one band correspond to the wild type homologue, which is the 5 kilobase, and 2 bands that correspond to 2 and 3 and 2 kilobases. So you would already know that your probe is binding to 3, not only 1 allele. So it's a very powerful technique, and with the usage of the bioinformatics and with the usage of the novel techniques like PCR and all that, this becomes even more and more powerful to detect diseases. And here I put for you a long list of, in fact, this is a very, very short list of the long list that we could predict based on simply using the RFLP technique. So all these diseases like sickle cell anemia, cystic fibrosis, Huntington disease, thalassemia, Duchenne muscular dystrophy, you can simply detect by this way. You have a specific probe designated to the gene that causes the disease. For example, if I quickly take the Duchenne muscular dystrophy, we know that the gene dystrophin causes this disease, so we design the probe that binds to the site specifically that causes where the mutation happens and the dystrophin is not functional, and then we can detect if the person is carrier for that SNP or not. We could also detect parasites and viruses, so we can tell if this parasite like more virulent compared to another one by simply comparing the genomes of the two. Like if we take malaria as an example, you take different versions of that, and you would do genotyping the same way we discussed, and at the end you have a specific probe to each type of the malaria, and then you can tell that this is more virulent, so we are detecting that it's there, but or it's not virulent, so this is another type. The same thing for viruses, and you can detect different versions of the virus, especially the mutated versions of the viruses. You can design probes, and you can tell that this is H1N1 or H2N2 and such. So the applications are really, really, really wide, and in principle, it's exact same procedure. Now, one thing I want to discuss with you, and I believe for the health sciences department, this is going to be important, is to identify the differences between personnel by identifying this difference in the 0.1% in their genome. And the way we can do that is by identifying tandem repeats. So these tandem repeats are in principle the variable tandem repeats. First, to start with, they are DNA sequences that happen to be repeated multiple times, one after the other. So why we say tandem? Because it's like ACTG, ACTG, ACTG that is repeated in a specific location on the genome. And this by itself is considered as polymorphism. But now let me just differentiate between two things. When we have polymorphism in terms of the gene expression, you will phenotypically see the difference. So when the gene, for example, doesn't produce insulin versus producing insulin, then you can then you can say that the polymorphism is that generation of insulin versus not generation of insulin. Or the eye color is different, so that's functionally clear that one gene is producing a black versus a brown color of the eye. But in the polymorphism we are talking about here is in the non-coding area of the genome. And therefore, the polymorphism is mainly by repeating those sequences, not necessarily that they will be transcribed into a protein or RNA. Maybe to a RNA, but not necessarily to a protein. So the polymorphism is the number of the repeats. Okay, just I want to stress on this, and it's in the non-coding area of the genome. And that's why it becomes really appealing to look for applications to check how can I use these VNTRs and look for genetic fingerprinting, so differences between individuals, and how can I identify someone that had left some DNA material in a crime scene, or whether there is a paternity test to make sure that this is the father of the kid or whatever. So after discovering the VNTRs, which are a little bit longer in size, there was the discovery of the short tandem repeats, which are kind of smaller in size or shorter. And that's why we call the VNTRs as mini satellites, and the STRs, we call them microsatellites. So I will quickly go to this. Okay. And okay, share. So this is the first DNA fingerprint that was done. So in principle, how does it work? So as I said, those variable number tandem repeats, they are different number of repeats. So within person A, you can see that he has 3 repeats on one of the alleles and he has 7 repeats on the other. While in B, you can see that it has exact same number of repeats on both loci. So in order to differentiate between A and B, what you need to do is to simply extract DNA from those 2 people, and then digest the DNA. The probe that you will use is going to be specific to those repeats. So the probe, as you can see, it will detect those 3 repeats on A or on B. And after digestion, you will have a different pattern of the DNA around this site. And obviously, you will have a mixture of all the DNA that comes from these 2 persons. So it's going to be a mixture of everything that you will eventually load on the electrophoresis gel. And after that, you will run the gel and you will get the smear because you have DNA of various molecular weight. After that, you're going to transfer this into a membrane. And you can see the bigger molecular weight is up, the smaller molecular weight is down, and you can see the patterns are different based on the digestion. And after that, you will apply the radioactive labeled probe to the membrane, and it will light up at certain bands. And after that, you will get a totally different restriction fragment profile in A compared to B. So directly telling you that A and B are two different individuals. So this was the first application that has been used to differentiate between personnel based on the restriction enzyme digestion profile. So if I stop sharing that and then I go back to share PowerPoint. Now, using the short tandem repeats is more powerful because now you can look at those differences in multiple numbers, but yet the differences could be really massive between two different individuals. So again, exactly like a variable tandem repeats, the short tandem repeats, they are polymorphic because the number of repeats you have. So it's the same thing. The only thing, they are shorter in size. And one thing that you have to keep in your mind, that within the non-coding part of the genome, these repeats are flexible. So they can become longer or shorter because the DNA polymerase, when you are doing the replication, is not going to be proofreading 100% compared to genes. So most of the time, these STRs can become longer or shorter, which give rise to many different alleles. And those different alleles, as I said, these are the number of the repeats. And therefore, that is another type of polymorphism that we are talking about. A concrete example I wrote for you here is in this sequence, for example, the CA has been repeated 13 times. So this can be a polymorphism that you can use to differentiate between two different people, simply by having a probe that is directed toward this particular sequence. And to give you an idea how we can use that in forensic studies, I'll come back to this video, but I will describe the steps again here for you. So first, you have to collect the sample. Let's say we have a crime scene or whatever. We want to know if this person was there. We have blood, we have saliva, we have whatever hair left. We need to take that sample and extract DNA from it. And if the sample is like a hair or blood, so this means it's not a lot of DNA there. So we need to amplify that DNA. So there is a step that you need to understand here that is added, which is amplification. And this will, here comes the PCR. I will be describing this, what is the PCR, in the next slide, but think that you need to amplify that DNA. Then after that, you would need to, again, digest and run on the electrophoresis as before. And then you would determine the sequences and you need to compare. So there's one thing you have to always keep in mind. If I'm suspecting person X was in a crime scene, this means I have to get the DNA material from the crime scene and I have to get a DNA from him so that I can compare these two. You need to have both components. So you need to get DNA from that person and the DNA that you are testing, and you run them side by side in order to do the comparison. Okay, so if this happens, then you can differentiate between the person, and you can say that this is the person who did this or not, or he was there or not, whatever the question is. And nowadays, the FBI have around 20 known loci, and I think it's not only the FBI, all the world, because this is the reference I used, that spread out over all around 80% of the genome, where we know that these loci can be used to generate probes that can differentiate between people. And therefore, if you get a hair or saliva or a blood or urine or a sperm, and you amplify that, and then you do the amplification step, and then you do the digestion step, on each of these chromosomes, like chromosome 2, you have a location that they call TPOX, which is a specific loci with a specific STR that can tell you that this is going to be almost unique between two different people. Now imagine you added this 20 different locations. So this is highly different from one person to another. Again, this on chromosome 3, the D3S1358 is highly different between two people. Then again, chromosome 4, 5, 7, 8, 11, 12, 13, 16, 18, and 21. So when you put all these together from one person compared to the DNA that you got from wherever, or the paternity test or the crime scene, it's almost impossible that these 20 look like the same in two different people, okay? And on top of this, you have the AMEL on X and Y that can differentiate between males and females. So after running this test and using all these probes, the combination of 20 probes, it's almost impossible that two people have exact same VNTR or STR. Okay, I will go to the video back. We're gonna watch together, and then I will take any questions.
I knew I was underwater. I just didn't realize I was this underwater. If you're stressed about your...
What is DNA fingerprinting or DNA profiling? Leicester University geneticist Alec Jeffreys developed a technique called DNA fingerprinting in 1985. It allows DNA samples from different people to be compared to look for similarities and differences. It's used for solving crimes and can also confirm if people are related to each other, like in paternity testing. Any two people in the world have 99.9% of their DNA the same. So this process analyzes the differences in the remaining 0.1%. This modern technology is called DNA profiling. It's a very sensitive technique which only needs a few skin cells, a hair root, or a tiny amount of blood or saliva. There are sections, or loci, of chromosomes, where instead of a gene consisting of a long sequence of bases, there are much shorter sequences of 3, 4, or 5 bases that are repeated many times. For example, these repeated sequences are called short tandem repeats, or STR. At these places on the chromosomes where we find these STRs, there are areas that vary in number of repeats. DNA profiling only looks at these STRs. A cell sample is collected. This could be from some blood at a crime scene, or a swab from the inside of someone's cheek, for example. The DNA is then extracted from the sample. Many copies of this DNA may be made using the polymerase chain reaction, or PCR. Special enzymes called restriction endonucleases are used to cut the DNA up into different size pieces. The DNA samples are then put into wells in a special gel called agarose for the process of gel electrophoresis, which separates the DNA fragments by size. The pattern is then transferred to a nylon sheet in southern blotting. And finally, the lines produced by the DNA samples from different people are compared. So let's have a go at using DNA profiles to solve some mysteries. An item was stolen in a burglary. A drop of blood was left behind by the thief. Samples of DNA were taken from 4 suspects and compared to the sample left at the crime scene. Which suspect is guilty? Here's a clue. Look for the one that is most similar to the one from the crime scene.
So you can tell right away who was there based on the fragment profile.
Pause the video while you decide. Suspect 3 is guilty. Can you see how the pattern of bands matches in suspect 3 and the crime scene? Their DNA is the same. Let's try another. A soldier has been killed in an explosion and has lost his dog tags that identify him. 3 soldiers are missing from their unit, so the army asks for 3 sets of parents for a DNA sample so that they might compare it to the soldier's DNA and therefore make an identification. Remember, the soldier will only share half of his DNA with each parent. So which set of parents is the soldier the son of? Pause the video while you decide.
So you have to be careful here that both parents has to be combined in terms of the bands in order to give you the profile of the soldier. And again, as you can see, we need the DNA that we are testing, and we need a comparable DNA. In this condition is going to be the parents in order to identify if they have the same pattern.
Parents C and D. As you can see, he inherited the first band from parent D, and the second, third, and fourth from parent C, and so on. So now you understand how DNA profiles or fingerprints are made and seen some examples of where they can be used. You can even interpret a DNA profile.
Okay, I'll stop to take some questions here. The difference between the two is just the how many times is repeated, and the shorter it is. So the STRs can be two base pairs that are repeated many times, 3 base pairs that are repeated many times. The VNTRs can be longer than that. Are there any questions up there that I didn't answer? So the difference in between the two is simply the number of the repeats and the length of the sequence. So I didn't want to really go very deep for you on this. I just give you the example here. So the STRs can be as short as 2 nucleotides. Like you see the CA is repeated 13 times. It could be a CA that is repeated like 20 times, right? Are these STRs the same technique also used in the cell line verification? Cell line verification? Did we mention anything about cell lines here, Aravind? The STRs? It's difficult to say, but I would assume yes. One thing you have to think about that cell culture, cell lines, they become more and more homogeneous with time. And it's very rare that people look at those STRs in the cell line that they are using, because these are not coding genes. Okay. Okay, so let's assume that you have a good idea about how we can apply the extraction of DNA, running it on a gel, and then really using it in a real life condition like matters us for paternity, for crime scene detection of the suspect, and then, for example, detecting if someone is disposed for a genetic disease or not, and differentiate between 2 different people based on their genotype. So if we move on, we'll come back to these steps. And as I said, one of the steps was to amplify the DNA with a PCR. So I'm assuming most of you know what is PCR, but let me go and describe it for those who didn't do much molecular biology and cell biology. So if we come back to the DNA replication, we said that in order to amplify the DNA, we would need to first unwind the DNA. And then this is a process that's done by helicases and topoisomerases. So these are specific enzymes to do that. Then we need the DNA polymerase, which will come and copy paste what each strand had. And therefore we get a duplicate of the DNA. So we will use some of the properties of the DNA replication in the PCR. But the difference between doing a PCR in vitro in the lab compared to DNA replication is one thing or a couple of things. First, DNA replication is taking place at 37 degrees in our cells. We can play with this in the lab up to any temperature we want. Then the DNA replication is taken all over the chromosome, and in the PCR, we decide the length of the fragment we want to amplify. In other words, if I want to amplify only this fragment from the T up to this guy here, all I need is just to design primers that fit on both sides, and then I will generate this fragment. So let's dive into the DNA amplification by PCR. So this would be the second component that we will mention. I mentioned before that restriction enzymes really made a revolution in the molecular biology, and then the PCR was also the second component that made a big change in the molecular biology, because you can amplify and modify any piece of DNA, any fragment of DNA, the way you like. It's very easy, very smooth, very elegant, and it can be done within 1 hour to 2 hours based on the experiment, and you can get these done. It's cheap as well, so it can be used to perform multiple analysis, and it's not going to be costly. Of course, there are several forms of PCR, and today we are just talking about the PCR itself. Later on, we will introduce more and more in the next lectures, particularly what we call quantitative or real-time PCR, that we can test the RNA analysis, but this will come in a lecture by itself. So in principle, in order to unwind the DNA, we simply heat it up to 95 degrees. Because in the cells, when the cell duplicate the DNA, it has the enzymes to do that. We don't have to use enzymes in vitro. All we need to do is simply unwind it by heating up the DNA to 95 degrees. And at 95 degrees, you will break the hydrogen bonds between the nitrogenous bases. Now, if you decrease the temperature from 95 back to 55, the DNA binding and re-annealing take place. But we can play with this by using primers. The primers, they have smaller sequences compared to the DNA fragments we are working with. And therefore, they have the ability to quickly bind to these large molecules. So in other words, if you have a DNA molecule that is 1 kilo base and you have a DNA molecule that's 20 base pairs, obviously the 20 base pairs will wind much faster to the DNA fragment compared to the 1 kilo base. So the 1 kilo base, it will take a longer time. Therefore, if we generate primers that are 20 base pairs, the moment we start decreasing the temperature from 95 up to, let's say, 55, quickly the primers will bind. And then we will add a polymerase that has the buffer that contains all the requirements in order to copy these two strands. So the polymerase needs magnesium, and we will have the DNTPs, the deoxynitrogenous bases, so that the polymerase can read and then generate a copy. And by the end of this cycle of denaturing, annealing, and polymerization, we will have already 2 copies of the same DNA. And if we keep repeating this again and again, you can have a massive amount of DNA. So here's an example. You can get up to 100,000 copies, or around 0.5 micrograms of DNA if you perform this in vitro. Now, what are the primers and how we design the primers is coming in the next couple of slides. We know that doing a PCR for short fragments of DNA up to 2 kilobases is easy, but if you want to do it for 40 kilobases or more, it's a little bit more difficult, but it's feasible to be done. Okay, so what do we need to perform a PCR? We will need a thermocycler. Obviously, that's the machine that will put the reaction at 95, then it will bring the reaction down to 55, and then it will bring it back to 72, where the polymerization takes place. So that's the machine. You will need the Taq polymerase, which will make the copies. So it's going to read the helices and perform the duplication process by reading a template and filling the gaps. You will need the DNA that will be the sample or the template that you will read from. You will need the primers, which are the shorter fragments around the size of the DNA that you need to amplify. And you will need DNTPs, the C, the G, the A, the T. Okay? So if we look at the process, this is a more in-depth explanation of the PCR reaction and the process itself. So it starts by denaturation, as I said, heating up to 95 degrees to make the DNA single-stranded. Then annealing so that the primers bind to their complementary side. So here they are giving you a very schematic example of a primer. The primer is much longer than that. It's 18 to 20 base pairs or even longer. But let's assume that this is a primer here. So this primer already bound and this primer is already bound. And then the Taq polymerase is going to come and complete this fragment synthesis on both sides. And then you get 2 copies. And you repeat this again and again and again. And think about it, that every round you do a PCR, you started, imagine, from 1 molecule here, you already ended up with 2. Then this molecule is going to become 2. So this is an exponential procedure generating tons of DNA of these specific DNA fragment. There is a YouTube video here. Let me see. Probably we will go back to watch. Now, if you go to a machine to perform PCR, you will see an example of the program. An initial denaturation step. So in order to have all the DNA denatured, we add all the time in the program an initial denaturing step, which is going to be 5 minutes at 95 degrees. That's before we start the cycles. And then we have to decide how many cycles, normally 20 to 30 cycles, depending on the experiment. Keep in mind that the longer you go, probably the polymerase will die. It's not going to be doing more than like 35 or 40 cycles. So if you want to go longer, you have to adjust the amount of the polymerase you use. And then the denaturation, the annealing, and the extension are discussed in the previous slide. They are the same. So this is repeated when it says 30 cycles. So this is repeated 30 times. At the end of that 30 cycle, we add a final extension so that all the molecules will be polymerized. We keep it at 72 degrees for 5 minutes. And by that time, you have amplified the DNA. And finally, if you set up the reaction, that's how it looks like. You will need, for example, a PCR reaction. The final volume is 25 microliter. 18 of that is water, 25 microliter. If we have the buffer 10x, it's 10%, so it's 2.5. We need MgCl2 because it's favorable for the polymerase to work with the MgCl2 as a cofactor. We need the DNTPs because those are the units that will be added to generate the DNA fragments. And then we need a forward primer and a reverse primer and the Taq polymerase. And these are the steps. Now, be careful that the extension step depends on the length of the DNA. So if the fragment you are amplifying is 2 kilobases, you would give like 2 minutes because it's like a rule that 1 kilobase per minute. Nowadays, some Taq polymerases are faster, but generally speaking, that is 1 kilobase per minute. So if your DNA fragment is 5 kilobases, you can't have only 1 minute extension. You have to have 5 minutes extension. But the denaturation, annealing, and final extension, they don't change, and the initiation doesn't change. So the only change in terms of the duration can be the extension time. Okay, now in order to design a primer, there are few parameters you have to look at. First, you have to get 18 to 24 base pairs in a primer. That's because before 18 base pairs, you will have a lot of unspecific binding. So the more toward 18 to 20, it's becomes specific to the DNA, and it will not bind somewhere else. The GC content, because of the hydrogen bonds and the annealing temperature, depends on that. It's recommended to be 40 to 60%. The start and the end, it's better to be G and C because there's a 3 hydrogen bond compared to 2 hydrogen bonds with the AT. The melting temperature, it's recommended to be between 50 and 60 because that's the favorable where the DNA re-annealing takes place. And the difference between the two primers, because we have one forward and one reverse, it's recommended not to be more than 5 degrees. So if one melting temperature is 55, it's ideal to have the other one 55, but you can play a little bit around 57 is okay, 58 is okay. The more than 5 degrees difference in melting temperature, it might not work. And then you have to be careful about the secondary structures, such as the primer can make a hairpin on itself, or it can make dimers with itself, or can cross dimer with the other primer. Those, there are a lot of different tools that you can use. So you have the IDTA here, and you have the primer 3. If you have a piece of DNA, you want to generate a primer, all you need to do is take that DNA, throw it there, and suggest what primers, and then you take those primers and test. And just to let you know, your next assignment is going to be designing a primer. So I have a YouTube for you here to check how to design a primer. And I have a YouTube here to check how to set up a PCR. So please go and watch these videos. And if you have any questions, come back to me on Thursday. By Thursday, I hope I will have the assignment already set up and sent out. So if you have questions, it would be great that you look at this part of the lecture and we can discuss. When will our assignment be due? It's going to be 1 week for this assignment because it takes less than 15 minutes. And you guys are so good in timing. Every time I get like 50 emails asking for extensions, and I ended up adding 48 hours for all of you. Okay, so any questions? We still have 4 or 5 minutes if you have questions about this lecture. The next lecture, we will start discussing the... Okay, so... Okay, since there was no questions, let me take a scenario here that I created for you. So this is a piece of DNA that I want to amplify or clone, but unfortunately, there is no restriction sites that I can use in the beginning or at the end. And the assignment is most probably going to be like this. So how can I clone this? I can take this DNA and I generate a forward primer and a reverse primer, taking into consideration the parameters I said, and you can go to the website and choose the primers from the program. And then, so I have added the two primers. But now the problem is that if I add the primers, I amplify the DNA, but I can still not be able to clone it. So most probably I have to add restriction sites on top of that. So then I add EcoR1 restriction site on the 5' end, and I add BAM-H1 site on the 3' end. So after amplifying this DNA, I can digest with BAM-H1 and EcoR1, and then it will be very easy to clone this gene into a plasmid. The plasmid should be also cut by EcoR1 and BAM-H1. Now, in order for the enzymes to cut, they cannot simply fly and cut. I have to add feeder sequences. So on top of that, I have to add this sequence, the GGGAAA on top, before the EcoR1, before the enzyme. So the primer tends to be almost 32 base pairs. And after getting this PCR, now the restriction enzyme cutting is very efficient because EcoR1 will come, will sit here and will cut, and the BAM-H1 will do the same. And then I will cut the plasmid with the same EcoR1, BAM-H1, and ligate it, and then I can clone this gene. So here you go. You can easily clone any gene from now on. Okay. I don't see the last assignment I submitted on Brightspace. So, Beta, did you email me? I think you emailed me and I asked the TA. I didn't get an answer. In principle, there is a log on the Brightspace. If somebody submitted, we directly see. So if you didn't get a submission done, this means most probably you did not submit your assignment on time. If you try to submit after Friday night, it's not going to allow you. So Anna, you are asking if the assignment is due 1 week from when you will make it available. Yes. It's going to be 1 week because as you can see, it's very easy. It doesn't take more than 15 minutes to design a primer. Okay, so you have another question. I submitted my assignment on time, but I don't see it on Brightspace. So Aliyah, again, this is something that I cannot control. I mean, everybody submitted their assignment to Brightspace, and it worked for 99% of you. So I don't see why it wouldn't allow you to submit it on the